Hello, everyone. Can you hear me? Great. So thanks a lot for the opportunity and, of course, for the prize. It's a great honor to be here. Let's start with a game, shall we? I'm going to ask you questions, and if you think the answer is yes, then please raise your hand, or hand, at least one hand. One hand is enough. Okay. So first question, as a warm-up. Do you enjoy your time here? Do you enjoy the conference? Okay, cool. Me too. So that's a good feedback for the organizers. Now, who would like to become a physician, a medical doctor from the audience? Many of you. Okay, that's great. Have you heard of the antibiotic resistance crisis and antibiotic-resistant superbugs? Many of you. Great, so you are also well informed. So you may have also heard that by 2050, antibiotic-resistant infections may become the leading cause of death if action is not taken. Why is it so? Because most of the medical interventions, basically the entire healthcare system, and even our food heavily relies on antibiotics. Humanity uses more than 100,000 tons of antibiotics every year. And now there are bacteria that become resistant to all of these. Have you seen the movie Semmelweis? Who has seen that? Please raise your hand if you have. Okay, cool. So you are really well informed. So you saw how miserable life was before antibiotics. Now the situation is that we may end up in the same situation if new antibiotics are not developed. So next game. Next question. You need to guess how many antibiotics are now in clinical trials. So clinical trials are the processes where drugs, including antibiotics, are tested on humans. It takes about 5 years, so it projects basically how many new antibiotics are going to be introduced to the market in the coming years. Okay, so to help you to guess, I brought you an example, cancer. Cancer is the leading cause of death today, with 10 million mortalities every year. So as a response, pharma companies have started or developing drugs and testing them in clinical trials. Specifically, more than 7,000 clinical trials are ongoing. So there is a huge hope that in a few years' time, cancer will be managed more successfully than today. Now let's see what's the situation with antibiotics. So there are 2 to 5 million deaths every year, depending on the way how it is estimated. Okay, now the question, how many antibiotics are in clinical trials? I give you 3 answers. So the first one, answer A, very bright future. Pharma already responded, and so therefore there are thousands of antibiotics in the clinical development pipeline in clinical trials. Answer B, moderately optimistic future. Or answer C, there are less than 50 antibiotics in clinical trials. So now, please raise your hand if you believe in the very bright future that pharma already responded and there are thousands of antibiotics. No one. Okay, so otherwise it wouldn't be a crisis, right? Who believes that answer B is the right answer? Some of you, very optimistic, or relatively, okay. What about answer C? Who believes that? You are very pessimistic, many of you. And actually, you are right, because the number of antibiotics in clinical trials is pretty low. Why big pharma stopped doing antibiotic development? The general answer is that because it's bad business. Okay, so it's really difficult so meaning that it costs lots of money, millions of dollars. And the investment is also very, very risky. So if resistance evolves against that new antibiotic and become widespread, then that new antibiotic's net present value, meaning the value on the market, goes down to zero. They won't be able to sell it anymore for high price. So their investment, lots of time and money, goes out of the window. What can we do about this? So maybe we could tell in advance, so we basically predict that how long an antibiotic is going to stay effective once introduced into the clinic. Obviously, we don't need a crystal ball for that. What we need are methods that accurately test antibiotic resistance evolution in the laboratory and does it at an early stage of drug development, so pharma companies can choose the right molecules for further development that are less prone to resistance evolution. So this is, this is what we came up with, a method that can mimic or model antibiotic resistance evolution in the laboratory. I also want, before we go into the details, I wanted to highlight that Dorina is a scholarship holder of this foundation. So when she started to be involved in the project when she was a first-year medical student, and therefore she became a co-author on this paper, so she could fulfill one of her major milestones during the scholarship, that is being a co-author by the end of the second year. So how does antibiotic resistance evolve? If we want to understand our technique, first we need to understand this. So the major way, the most frequent way for antibiotic resistance to evolve is horizontal gene transfer. Horizontal gene transfer is a process when a bacteria, an already resistant bacteria, provides a piece of DNA to another bacteria that is sensitive to the antibiotic, and therefore it becomes resistant to that antibiotic. Okay, so this piece of DNA called an antibiotic resistance gene. It's like you have blue eyes, and then you touch someone, and suddenly he or she will also have blue eyes. How cool is that, right? However, everything has a price. For example, bacteria do this horizontal gene transfer instead of sexual reproduction. So I believe we are much better off. But anyways, going back to science. So if resistance evolves with horizontal gene transfer, then how can we estimate how long an antibiotic stays effective? In general, we believe that we need to estimate how many antibiotic resistance genes are out there in the environment. Okay, let it be hospital environment, soil environment, or farm animals. If the size of the resistome, so basically the sum of all the resistance genes that can compromise a certain antibiotic that is available for the target pathogen for uptake, then that antibiotic probably will be compromised very quickly. Ideally, a good antibiotic shouldn't have any resistance genes out there. Okay, so this is how the technique works, how this technique estimates this feature. So first we collected environmental samples from different antibiotic polluted environments like hospitals, the gut microbiome. The human gut is a huge reservoir of antibiotic resistance, and antibiotic polluted environmental, like soil samples in India, where antibiotic concentration close to antibiotic factories is so high as in the blood after antibiotic is injected, because antibiotic is an environmental pollutant in these environments, so there are lots of bacteria that try to adapt harboring lots of resistance genes. So we extracted the DNA out of these environmental samples from the bacteria that live there, generated DNA fragments out of those, more than 10 to the 7th fragments that covers thousands of bacterial genomes. And then using genetically engineered bacteriophages, tiny viruses, we injected this DNA library into cells. So each cell expressed a different library member, a different gene. Then we cultured these bacteria in the presence of the different antibiotics, and those colonies or cells that survived, they contained a resistance gene from the environment. So this whole process mimics how horizontal gene transfer works in real life, in nature. At the end, we read out the DNA, the identity of the antibiotic resistance genes by DNA sequencing. So here I just show you the last step, how the bacteria are cultured. Here you can see two Petri dishes. One contains only the bacteria that grows at lower antibiotic concentration, and in the presence of increasing antibiotic concentration, it stops growing. This yellow line represents the inhibitory concentration when it doesn't grow anymore. Okay. So in the other plate, what you can see that cells, after receiving environmental DNA, started to grow even at higher antibiotic concentrations. These are the bacteria that got an antibiotic resistance gene against that specific antibiotic. So we repeated this experiment 156 times to screen basically 3 environmental sources: Indian soil, human gut microbiome, and clinical microbiome, in the presence of 6 old antibiotics and 7 recent antibiotics that just came out from clinical trials. Okay, and made these experiments in 4 different target pathogens, 4 different antibiotic-resistant species. And then the surprise came when we summed up how many antibiotic resistance genes were present for the specific antibiotics. Each antibiotic is 1 column here. We realized that recent antibiotics that just finished development have as many resistance genes in the environment as antibiotics that have been used for decades. Okay, so you can see, for example, gepotidacin, a new antibiotic class, new mechanism of action. Still, there are more than 20 antibiotic resistance genes in the environment that can easily compromise this antibiotic. So these antibiotics won't take long. Will be compromised very quickly. So, meaning that even those that are in development are not the right ones. We need better antibiotics. So now what we are doing now, we are testing all the antibiotics in clinical development, try to prioritize them to spot which are the best ones, because we need to focus only on those and not others. Finally, I'd like to show you where we work. So we work in the Biological Research Center Szeged, which is the biggest life science research institute in Hungary. Here is the specialized laboratory where we work safely with these pathogens in high biological containment. And here you can see the group. There are lots of young scientists and students. 2 senior or National Academy of Sciences students works there who are just a few years ahead of you. So if you keep pushing, then in a few years' time, you can join the research group, whichever you would like. Thanks a lot for your attention.